Okay, so today I'm going to talk about the rest of the antigen presentation material that I did not talk about before break. I hope everybody had a fantastic break. And you should have gotten an email from me that we're going to do the paper discussion of the Golder paper on Friday with this. So remember, you'll have the discussion questions due. Also, the MHC antigen presentation problem set is due on Monday. That way, you have some time between when we talk about some of the material today and when that problem set is actually due. So you can come ask questions, and please do so if you have any. One other thing is that my list of things that I told myself, oh, I'll get that done over break, was a little ridiculous. So I'm not done with the exams. I apologize. After class today, I am first going to eat lunch, and then I am going to sit in my office and finish them. So they are in fact the next thing that I'm finishing, but I do not currently have them for you today, and I apologize for that. So today, antigen presentation, part 2. If you recall, we are really thinking right now about the process by which the pathogen protein that's shown here in red gets degraded and loaded onto an MHC molecule. And we went through the steps of this process for MHC class 1 before break. I typically break this into these 6 specific steps, and I listed the 6 steps for class 1 up on the board, which you can also see the class 1 process summarized here. Remember, class 1 has its heavy chain, the alpha chain, along with the smaller beta-2 microglobulin, or beta-2M, that's not covalently attached. And here you can see the peptide binding cleft. All nucleated cells are presenting on class 1, and they're presenting to CD8 positive T cells. The peptides are coming from the cytosol, usually by infection. Those peptides are then tagged with ubiquitin and degraded by the proteasome. We then will move those peptides from the cytoplasm into the ER using the TAP transporter. That MHC class 1 molecule was held next to the TAP transporter and is held by chaperones, particularly Tapicin, to sort of allow all of this to work and allow binding to happen. And once that binding happens, display is constitutive, so sort of automatic, no effort needs to go into making that process happen. And after the video that you saw at the end last time, I'm guessing you'll never forget TAP. As I introduced last time, immunologists often think about the cell in this way in terms of the yellow areas versus the light blue areas, sort of the cytoplasm and across a membrane from a cytoplasm. So we've talked through class 1, and class 1 is really the way that proteins from the light blue area get presented. We're now going to switch gears to talk about the steps for class 2, and class 2 is really the way that peptides from the yellow areas are getting presented. So things that are in the biosynthetic secretory pathway getting ready to be secreted, or things that have been brought in some kind of vesicle. So here is class 2. Remember, only certain specialized cells, the professional antigen presenting cells, are presenting on MHC class 2, which are B cells, macrophages, and dendritic cells. Class 2 has these 2 proteins that are sort of similar in size, both with transmembrane domains. We're presenting to a CD4 T cell. One thing that I also mentioned about the class 2 complex that will come up today is just like with class 1, we don't really get proper folding until we have both proteins and a peptide in the cleft. But when we do have both of the MHC class 2 alpha and beta chains and a peptide in the cleft, this complex is incredibly strong. I mentioned to you that it is SDS stable. So in fact, things that we usually use in biochemistry to denature proteins do not denature this complex. And so the chains with the peptide in between stay together when all other proteins get like totally linearized. So it's a very, very, very stable complex. And as we've seen in a couple different ways, it is presenting on class 2 peptides that are from the endocytic pathway. We can divide up the steps here exactly the same way into exactly the same 6 steps that we saw before. Again, if you look at some of the steps from class 1, some of them you're like, that's really lame. Why did she give that its own step? And the answer is because something interesting happens there for class 2. And there are similarly going to be some steps that you're like, that is a dumb step for class 2. And it totally is, but it's interesting for class 1. So the first thing that we can think about is how exactly proteins get into this endocytic pathway, how we acquire the antigens. With class 1, this was about being infected or having made the protein in the cytoplasm. And the cell really didn't have to try to make those things happen. In the case of class 2, however, the cell has to actively do something to acquire the antigen. The cell has to try to acquire the antigen. There are 2 ways that the cell can acquire the antigen. One of those 2 ways is shown here. And so this is phagocytosis, which we've talked about previously. You can see that we may have some kinds of receptors on the surface of the cell that may bind to the microbe. Now we're going to internalize that microbe into a little compartment because that compartment was made in a process of phagocytosis. We call the compartment the phagosome. And that phagosome will eventually fuse with the lysosome, will become acidic, and will degrade that microbe. This is just like what we saw with neutrophils and macrophages degrading microbes and killing them. It also just so happens that this is going to be the beginning of the MHC class 2 pathway. And so phagocytosis is something we've already talked about. And it is one of the ways that we acquire antigen for class 2. It is not the only way that we can acquire antigen for class 2, however. If you think back to, well, I guess, yeah, okay. What I want to show you on this slide is that there are lots of ways that cells can internalize things in terms of cell biology. So you're seeing a few different ways of cell internalization here, like clathrin-coated vesicles and other kinds of vesicles, compared to phagocytosis. If you look at this sort of area that's being made by phagocytosis compared to the other kinds of internalization, what do you notice about it? It's huge. It's way, way, way bigger. There's a lot more stuff going on with the membrane. There's a lot more going on for that phagocytosis to happen. Here is actually showing another image where you can actually see how much the plasma membrane had to deform in order for the cell to do phagocytosis. When phagocytosis happens, there's a ton of cytoskeletal rearrangement with actin and things like that. Phagocytosis is a very, very active process, and it's a very special kind of internalization. I mentioned to you earlier in the semester when we were talking about innate immune responses that in fact, in your body, there are really only 3 cells that we think of as doing a lot of phagocytosis. What were the 3 cells that I told you as really being the cells who know how to do this complicated process? And what I hope you see here is that it's like a special complicated process. I said there are only 3 kinds of cells that really could do it. Yeah. Macrophages, absolutely. Yep, Alexis. Not so much with the mast cells. Yep. Neutrophils. Macrophages, neutrophils, one other one. I may not have highlighted it a ton. The other one is a dendritic cell, which is related to a macrophage. Okay? So those are the 3 that can do phagocytosis. Let's compare that to the types of cells that can present on MHC class 2. You can see again, it's 3 kinds of cells. But is it the same 3 cells? What do you think, Josh? Yeah, here, B cells are presenting on MHC class 2. They weren't one of those cells we just listed that could do phagocytosis. And neutrophils can't do class 2, even though they can do phagocytosis. So this is one place where we get a distinction a little bit. What you should realize from this is there has to be a way to get antigen other than phagocytosis. Because how else can B cells do it? If B cells can present on class 2, but they can't do phagocytosis, there's got to be some other way acquisition can happen besides phagocytosis. Here you're seeing what happens when something gets phagocytosed. We make the phagosome. Eventually that phagosome fuses with the lysosome. For many of the other types of internalization, we make a smaller little vesicle here. And we call this endocytosis, and we usually call the thing an endosome because we came in through endocytosis. And eventually, the stuff in the endosome ends up in the same place that the stuff in the phagosome ends up. They all end up in the lysosome. But it's just sort of how big was the thing that was internalized. B cells will do this process a little differently than will macrophages and dendritic cells. If you recall, B cells have a B cell receptor on their surface that will bind to a specific antigen, and you can see that happening here. And we talked about that potentially activating the B cell. We didn't talk a ton about what that does to the B cell, but it does a bunch of things to the B cell. We'll see them. But one of the things that it does is that when that B cell receptor binds antigen and gets a signal, that tells the B cell to internalize that B cell receptor, to pull in that B cell receptor along with anything that is bound to it. Sort of like, hey, you caught something on your fishing pole line, pull it in. And so you can see that B cell receptor gets internalized. This is a specific type of endocytosis. So I told you endocytosis is when you bring in a sort of smaller thing. This endocytosis happened because the receptor bound some stuff. And so the name for this special kind of endocytosis is receptor mediated endocytosis. So B cells don't do phagocytosis, but they use their B cell receptor to do receptor-mediated endocytosis and internalize the antigen that way. And so you can see we make an endosome. It has both the B cell receptor and it has the antigen. You can notice here it tells us that the endosomes are more acidic than outside the cell, and I'll mention some more about that acidity later on. But you can also notice that when we get to that level of acidity, the antigen dissociates from the B cell receptor, and the cell can basically recycle the receptor back to the surface and do stuff like, I don't know, antigen presentation with the antigen that came in. So this is kind of how that acquisition process looks in B cells compared to in macrophages. Once we have brought something in, either through endocytosis in these endocytic vessels, or if it was phagocytosis, we got it in this secretory pathway. We got it inside one of these vesicles, all of whom are sort of interconnected with one another and who are sharing components or sharing stuff that's inside them, cargo, with little vesicles going in between. And so we're in the pathway. If you remember, in the case of MHC class 1, that was a big challenge, was how do we get the peptides into the pathway? We had to have that special transporter TAP to get the peptides from the cytoplasm into this pathway. Oh my gosh, we're already in the pathway for this one. So in some ways, it's like, oh yeah, we're good. This seems pretty straightforward. And to be honest, this is one of the places where one of the steps get weird. You don't really have to do anything special to tag things for destruction in the pathway, because this pathway pretty much always leads to the lysosome. So just like by being in this pathway, you're tagged. So again, there was a nice tagging was like a thing here. Now it's just like, yeah, whatever. You're in the secretory pathway. I told you last time that the cell has two ways to destroy stuff. It has two sort of trash cans, two ways to destroy proteins. And oh my gosh, they line up to either for the cytoplasm or for the secretory pathway. And the trash can of the cytoplasm is the proteasome. And that was the one that we used for proteolysis in class 1. So it may not surprise you that the thing we're going to use for proteolysis in class 2 is the trash can of the secretory pathway. So I moved that late last night, and I forgot that I moved it. You can see some of the names of some of the parts of the secretory pathway here. So, for example, sometimes we'll see something that we refer to as an early endosome. That's usually because it was made just after endocytosis or just coming from the Golgi. That can eventually become things like a late endosome. That might happen because of fusion of the early endosome with other things, or it might actually be a maturation of the early endosome. And eventually we can get to the lysosome. There are some other cell biological compartments sometimes that people talk about when they talk about this process. So as you will see moving forward, the lysosome is going to be the place where we really degrade stuff and do proteolysis. And you can easily imagine if I have a cargo in my endosome, my early endosome, see there's my triangle cargo. Eventually it's going to be inside of my late endosome, and it's going to be eventually inside my lysosome, and I'm going to degrade it, and we're going to live happily ever after. If I want to have a transmembrane protein, like if I'm making something that's in the plasma membrane, it's eventually going to be in the early endosome membrane, like that. And eventually it's going to be in the late endosome membrane, like that. And eventually it's going to be in the lysosome membrane, like that. And you can imagine if the lysosome is a good place for degrading, that we can degrade the stick part of this pretty easily. But then it's kind of hard to degrade that like triangle-y part because it's not inside the endosome. We've since learned that sometimes those endosomes can actually internalize themselves. And so you can have an endosome that actually brings little pieces of itself inside. And so you get these little internal vesicles. And so here you can see there is a vesicle being budding off of an endosome and going inside the endosome. And so you get this thing that has like little vesicles inside the vesicle. We refer to that as a multivesicular body. And multivesicular bodies are a way that we can allow those cell surface proteins to get degraded. And so you can see as we learn more and more about this process, we learn that there probably might be more types of compartments with more fancy names that we hadn't described before. The one that sort of a lot of people will talk about is the multivesicular body. You can actually see a multivesicular body here as well. And so just note that these are some of the compartments that we might see. This will come back later on today. So here we can see, oh my gosh, acquisition. We've done endocytosis or phagocytosis. And then we didn't really have to do anything to sort of tag this antigen. It's in the secretory pathway. And just by moving through the secretory pathway, it is going to get proteolyzed. Because as we go through this process from early endosome to late endosome to lysosome, we have more and more acidity. So we have decreasing pH throughout that time. And as we get more and more acidic, that just already automatically starts to denature the protein. So you can see we have acidification of vesicles. In addition, when we get to that final lysosome, which you can see here, has like a pH of 5, so it's really acidic in the lysosome, a whole bunch of enzymes start to be able to act. They're all enzymes that only can act when they are in an acid environment. So these include nucleases, proteases, glycosidases, lipases, phosphatases, sulfases, phospholipases. Basically, anything you need to degrade biological material, you have in these compartments. And once the compartment is acidic, we get degradation. And so the degradation here and the proteolysis happens due to just being in the lysosome. Please note that this is the lysosome, which is different than the enzyme you saw earlier in the semester, which is lysozyme. I've seen people get that one confused a few times in my life. And again, it's sort of like once the proteins get into the lysosome, which is their like place they're going, if they were endocytosed or phagocytosed, they're getting degraded in this acid environment with all of these different enzymes that are activated by acid and allowing degradation. So in a lot of ways, some of the things that you can think about with class 2 seem pretty straightforward. We are phagocytosing endocytosing our material, and we're already in the secretory pathway by definition. Hooray! And just by being there, we're going to get degraded. Hooray! So we didn't really have to do much like that we weren't already doing as general cell biology. And if you remember, we had this problem in MHC class 1, which was this problem of delivery. And the delivery problem was the fact that if we wanted to put MHC class 1 on the surface of the cell, it had to go through the secretory pathway. So it had MHC class 1 as a protein itself had to be biosynthesized in the secretory pathway. And we had this whole problem of how do we get our cytoplasmic peptides into the secretory pathway. We need to use ATP in this transporter to make it happen, right? Well, here you're like, look, it's magic. It's real easy. It's not hard. There's no delivery problem. But there is a delivery problem. And this figure from your textbook kind of underscores or overlooks a rather large problem that comes up in this situation. So this is a version of an image from your textbook that I have tried to modify. My modification is still not perfect. The way that they drew this, like they could draw it in a way that would be so much better, and they don't. And it just makes me so mad. But here you can see a cell. And note that that cell is presenting on class 1 and class 2. Recall, all nucleated cells present on class 1. So everybody's presenting on class 1. If you're presenting on class 2, if you're a B cell, a macrophage, or a dendritic cell, you're doing both. You're not doing just one or the other. You happen to be a nucleated cell if you are also an antigen presenting cell. So you're usually doing both at the same time. And they put a line down the middle in this figure. There is no line. I wish they drew it without the line, because that would make this perfect. So here's the cell doing its class 1 business, right? It's biosynthesized class 1 in the ER. We're going to shoot peptides into the ER using TAP, and then we're going to present those class 1 peptides. Well, class 2, we also want to present on the cell surface, which means it also has to be biosynthesized in the biosynthetic secretory pathway. It also has to be translated at the rough ER and made in the ER, which means at the beginning of this process, it's also sitting here in the ER, just like class 1 is. So what's the problem? Only they drew this without a line. It would be so much more obvious. Yeah, Josh. Mm-hmm. Yeah, now instead of getting peptides towards the MHC class 1, we're getting the MHC class 2 towards the peptides. Yeah, so first of all, we have to somehow get this MHC class 2 to the peptides, right? So it's delivery, it's just like the other way. It's delivery of the MHC to the peptides. But there's another issue with that. There's another reason why this is actually really a hard problem. Because they do a lot of oversimplifying in this image. What do you notice about that MHC class 2? Yeah, Andrew.
Because it's also this extracellular, and then it has to like get inside the cytosol to do all like peptides and
No, so it's going to do all of the things that it needs to do in the vesicular transport pathway. So we don't need to access the cytoplasm. Yeah, Emma. So it's a transmembrane protein, absolutely. But we can handle that. The cell can handle trafficking it and moving it. That's sort of basic cell bio. What do you notice right there? What is it, Andrew? There's nothing there. It's empty, right? And this figure shows, here's class 2 sitting there empty, sitting here empty all this way, sitting here empty, 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 until it gets to these peptides. Does that strike you as odd in any way? It's in the ER where there are transporters shooting peptides in. And yet somehow it's supposed to stay empty? The big problem here is how the heck do we get class 2 to the peptides without picking up some other peptide earlier on? How do you get a class 2 that's like ready to bind to get to these peptides? How is it that it didn't just steal all class 1's peptides? Because there's no line there. Those little red dots could diffuse around here and could get into class 2. Just, some of them probably fit class 2. Because we weren't like trying to make perfectly class 1 size peptides. We're just making some peptides and shooting them in. Some of them would fit class 2. And so the big problem here is how do we deliver class 2 to the peptides? And how do we deliver class 2 to the peptides without something else already in it. So this, again, this image from your textbook is just like, magic, done, easy. But that's really what the problem is. And so there is one protein that does a lot of work here. And this protein actually solves two different problems. And they're both problems that you guys have mentioned. This protein is called the invariant chain. And the invariant chain is biosynthesized with MHC class 2. What you can see is that it has a transmembrane domain and sort of comes up along next to class 2, and then has a little part that binds in the peptide binding cleft, and then has a little part that comes up here. So this whole green thing, including the purple, is invariant chain, sometimes abbreviated II. And the invariant chain usually holds a bunch of class 2s together, so it usually is a complex that looks a little bit like this. An invariant chain, like I said, serves two jobs. Different parts of it are serving different jobs. This part down here with the transmembrane domain, sort of this piece of the tail here, it solves the problem that Emma mentioned. It actually gives the instructions on how to do this movement. It's sort of like the address label that says move this thing into this place where the peptides are. And so that tail part of invariant chain moves class 2 where it needs to go. And so part of what invariant chain is doing is helping deliver class 2 to the peptides. The other thing that you can see invariant chain doing is that it actually sits across the peptide binding cleft and physically blocks the peptide binding cleft. So other peptides from other places cannot get picked up. And so you can see sort of this same process happening here. See, this textbook doesn't put a line. So it's a little more obvious that the peptides could go on to class 2, but fortunately, class 2 is protected by the invariant chain. And the invariant chain helps bring class 2 where it needs to go. As the invariant chain and class 2 are moving through these different parts of the pathway, we're getting sort of more and more acidic. We're not all the way acidic, like lysosome pH 5, but we're getting more and more acidic. And as time goes on, invariant chain gets degraded more and more until there's only one little piece left, and it's just that little piece that's blocking the peptide binding cleft. That one little piece is known as CLIP, or the class 2 invariant peptide. So basically, we're going to take class 2, we're going to have CLIP in the peptide binding cleft. We're going to have other parts of the invariant chain help move. And eventually, we're going to get class 2 plus CLIP into the place with the peptides. So now we have actually delivered a semi-empty, not actually empty, class 2 here to the peptide, so we can allow for binding. And this is kind of important for a couple reasons. And this also is going to lead to sort of the next part of this binding. I told you something about MHC class 2 with a peptide involved earlier in terms of biochemistry. What did I tell you about class 2 when it has its peptide in the peptide binding cleft? Yeah, Ashley? It's a very, very strong, stable complex. So if MHC class 2 had picked up a peptide it wasn't supposed to way back here, that would be super strong and it would be really hard to like get it out because it's so strong, right? So you got to make sure that does not happen. But that also leads us to another problem. Okay, if we look here, here's MHC class 2. It's coming in my conveniently boxed out, covered up area. It's going to come to the same place as the peptide, and we want to put the peptide in the cleft. What's the problem? Yeah. CLIP is there. CLIP's in the way. And oh my gosh, CLIP basically is a peptide, class 2 invariant peptide. It's bound really strongly there. It's another situation where you're like, oh yeah, it's already like real strong bound there, which is good. It was like doing its job of keeping stuff out. But now you got to figure out a way to get this really strong binding partner out. And this leads to this other protein that helps make this happen. And so this other protein that helps make this happen is involved in binding. And this protein, it's weird because students forget the name of this protein all the time, but it's like their favorite protein. They love what it does, they just can't remember its name. So we're going to both remember the name and the fun thing it does. And I'm not going to do this demo that I have done in past years because Dunaway yells at me for this demo, and I'm not going to do it. But it's a really good demo. So at this point, you can imagine we've got MHC class 2 with CLIP in its cleft, right? And we're going to deliver into the next compartment where the peptides are. In this next compartment, there is also going to be another protein. And that other protein's job is to remove CLIP from MHC class 1. The way, or from MHC class 2, I'm sorry. So the MHC class 2 is now empty and other peptides can potentially bind. The way this protein is known as, in the case of human, it's HLA-DM. In the case of mice, it's H2M. So it is encoded in the MHC region. People were sort of confused about what it was for a while, but it's like a helper for the class 2 process. And the way that HLA-DM works is HLA-DM basically comes up next to MHC plus CLIP. There's my MHC plus CLIP, this chair, and the little Apple pencil is CLIP. And HLA-DM basically does this. It literally makes a mechanical shove. It is a physical interaction and just shoves it until things fall out. I used to do this where I would like shove a student, and then Dunaway yelled at me, and it was hilarious, but we're not going to do that. You would totally remember it though. So we have this protein sitting in the same location as the peptides. We deliver MHC class 2 with invariant chain. We degrade that invariant chain, so eventually there's just CLIP left. Now we have our MHC class 2 in the same place as the peptide. And HLA-DM mechanically shoves class 2 to dissociate the CLIP from class 2, allowing some other peptide to come in. And in fact, that process will happen over and over again until a peptide is bound with such high affinity that DM cannot knock it off. And so that's part of the way that we get such a strong complex, is because it's actually unable to be knocked off by DM. So one of the things that you might have noticed as I've been talking about this is I've just said in the compartment with the peptides, or in the place with the peptides. I haven't like said, and we are now in the late endosome, or we are now in the lysosome. Like I haven't called out a place. And so immunologists have spent a lot of time trying to figure out the place for this to happen. you know, it's been a huge amount of work. People have done lots of different experiments to try to figure out the answer. And their answer is generally, I don't know. Basically, none of the experiments quite match up. And so we've made up our own compartment. And so the compartment where MHC class 2 loading happens is the MHC class 2 loading compartment. Because it doesn't seem to match up really well with any particular known compartment. And so one of the things that this has also told us is that we need to learn a lot more about these compartments in cell biology if we're going to actually be able to name this compartment and figure out additional details. When we look under a microscope at the MHC class 2 loading compartment, which is shown right here, it looks like a multivesicular body. It seems like a multivesicular body. Almost everything about it seems multivesicular body. So we may, in fact, find out that that's what it is, but it's still not really clear, and it's still at this point just we call it MHC class 2 loading compartment because we can't officially define it. And so then we can get into the last part of this, which is display. When we were in the case of MHC class 1, remember that display was constitutive. So basically, once you had a peptide bound into MHC, you threw it up on the surface. It was automatic. You didn't have to do anything. Every cell, all the time. Right now, all of your cells are automatically displaying stuff on their surface. It's a little different in class 2. In class 2, the cell will actually hold those MHC class 2s with peptides until they get a signal to display them on the surface. And so it is a regulated process here, unlike MHC class 1. What we found is that those little vesicles, the MHC class 2 loading compartments, seem to travel along microtubules and eventually get shifted up to the surface of the cell when the cell is ready to present. You can actually see this happening here. So here is a dendritic cell. So this, like, with the little jaggedy edge thing is the dendritic cell. This is a T cell. So the dendritic cell in the green is actually the MHC class 2. And so you can see originally the MHC class 2 is kind of like near the middle of the cell. But when the cells actually interact, the dendritic cell actually extends this little track of class 2 out to the T cell on microtubules. And so you can see it actually extends out the MHC class 2, kind of presents it right there to the T cell, and eventually even retracts it back. So here you can see a dendritic cell that is not activated, and you can see it just has class 2 kind of wherever. And then if you activate that dendritic cell, it puts all of that class 2 all up around the surface in order to present to cells. So this is an activatable process that is happening and occurs through transport on the microtubules. This paper that I have on the right, I always feel bad about it. I always feel bad about this, but I can't not say it. So Marianne Bose did this work, and she actually made up a name for this process. And I really hate it because it's not a word. So I'm going to tell you what it is, but then we can, if you really want to write it as, but I can't even write it on the board because it's not a word. And so according to her, this process is called tubulation, where the microtubules bring those things to the surface. And it's not a word, but this whole paper talks about tubulation and the importance of tubulation bringing class 2 to the surface when it's activated. And so that's sort of the important piece of class 2 display. So here you can see the general overview of both class 1 and class 2. This is the real version of the one from your textbook with this line in the middle. There's really no line. And you can see these steps for both of these processes. So I've got a few different things in the slides sort of remaining today. I have this slide and the next slide, which are really just compare and contrast of class 1 and class 2. So I'm going to mention those really quickly. I also have some kind of other details about presentation, sort of other aspects of presentation here, and I have a couple of like sort of problems that we can work through. So I want to just show you that I have this information. This is everything I basically told you about class 1 and class 2 in terms of compare and contrast the structure and function, as well as things like which T cell they present to, which cells do it, all of those kind of things. So these may be very helpful for your studying. All right. So there are two weird situations that I want to tell you about. I'm actually going to do this in a slightly different order. So remember that our whole discussion right now of MHC and MHC presentation is in service of thinking about T cells. The whole goal here is that we're talking about the antigen that a T cell receptor binds. And we're going to be talking about this paper about antigen presentation on Friday, and then on Monday we're going to start thinking about the T cell receptor again. And so really, all of this has been in service of the T cell receptor and T cell responses. There's one little, well, there are a lot, but there's one flaw in the plan I want to introduce to you now. And I'm going to flip to this issue first. This one. Okay, so this is 34 if you're looking at slides. So here we have our T cells, either CD8 or CD4, binding to MHC, either class 1 in the case of CD8, class 2 in the case of CD4, and that activating the T cell to make a good response. We can have basically any cell of the body presenting on class 1, right? So if I have an infection, I don't know why it is everyone uses the same example for this, and I'm going to use that example too, but there are other examples. If I have a hepatitis virus infection, hepatitis infects the liver. So in theory, that virus is in liver cells, and liver cells can present on MHC class 1, right? To turn on CD8 T cells. Like, this all makes sense. Is it possible for me to actually make a MHC class 2 response to that virus? As you will learn later, CD4s are actually kind of important, even if they weren't in my dissertation. Can I make a class 2 response to that virus? And if so, how? So in reality, the answer is yes. Either some parts of the virus get phagocytosed by a dendritic cell, or the liver cell dies and its parts get phagocytosed by a dendritic cell, right? So you can imagine a dendritic cell is going to get some parts of this either from virus just being around or from a dead cell and present on class 2, right? That doesn't seem like impossible. If you didn't want to imagine this happening with the liver cell, you could also imagine the cell in my arm right here where I got boosted this weekend. So you can imagine my muscle cell can present on class 1, and perhaps some dendritic cell could do some maybe a muscle cell dies, dendritic cell does some phagocytosis, I present on class 2. Seems great, right? Hooray! little bit of a problem here. In that, if you want to turn on a T cell for the very first time, like that, that first T cell that's going to respond to my hepatitis virus, or the first T cell that's going to respond to my vaccine. If you want to turn on a T cell the very first time, you really need a dendritic cell to do it. Dendritic cells are so good at activating T cells. Dendritic cells are amazing at activating T cells. Part of the reason is because they have all these proteins on their cell surface that can help with the activation. So dendritic cells are like the all-stars of turning on T cells. Part of the reason I haven't said a ton about them before this point is because their biggest job is turning on T cells, and so we can't really talk about them until we're talking about T cells. So, okay, now let's think back about my hepatitis virus. Hepatitis virus infects cells of the liver, and we'll say only cells of the liver. We might be worried about my CD8 T cell response here, because who's presenting on MHC class 1 to a CD8 T cell in this situation? Yeah, the liver cells. And the liver cells are kind of sucky at class 1, at least in comparison to dendritic cells, who are just absolute all-stars, right? So it's not the best possible way of turning on CD8 T cells that it could be. And so it turns out that dendritic cells have a special function. One thing I will also say, I haven't said this before, everything I've told you about class 1 and class 2 so far sometimes can be referred to as classical, like the classical class 1 and classical class 2 pathways. Dendritic cells are special in that dendritic cells can do some extra things. And dendritic cells have this ability to phagocytose things. So basically, to acquire things like they're being acquired for class 2, but then to allow those things to leak into the cytoplasm so that they can present anything they phagocytosed on both class 1 and class 2. So that dendritic cell, even if a pathogen doesn't infect them, even if the pathogen didn't first come to a dendritic cell can present it on class 1 and class 2. And it's really just about things leaking out of the phagosome into the cytoplasm so that dendritic cell can present on both class 1 and class 2. This process is known as cross-presentation. And so in reality, in making real immune responses, cross-presentation is incredibly important. And it's just something that dendritic cells can do just because they are such absolute all-stars. Let's see. Yeah, okay. So the other thing I want to tell you is that I mentioned before that T cells only bind proteins, and they only see proteins as an antigen. And that was in contrast to what we saw with antibodies that could bind to proteins or nucleic acids or carbohydrates or lipids. That is true of normal T cells. We have since found that there are some weird T cells. And one of the sets of weird T cells doesn't bind to protein antigen. Instead, it binds to lipid antigen. And so there's also this process where we can see lipids getting presented in addition to proteins. You can see a lipid on the left here. And what I want you to notice about the lipid is that there's some kind of head group, which is a hydrophilic head group, and then there are these long fatty acid chains. And that's basically what you need to know about the chemistry of lipids for this. Lipids can also be presented. Lipids are, and they eventually turn on weird T cells, which I'll tell you more about them in a second. The molecule that does the presentation is not MHC class 1, and it's not MHC class 2. It's its own special molecule that does presentation. And the name of this protein is called CD1. So CD1 presents lipids, sort of analogously to class 1 or class 2 presenting proteins. When we look at the structure of CD1, this is what we see. So we will notice that CD1 has one large chain that sort of can vary in a few places, has one transmembrane domain. Here you can see the lipid binding groove. And CD1, in fact, pairs with beta-2 microglobulin, the same partner that class 1 uses. And if you actually look at the crystal structure of CD1, which is in this like pink and purple, and the crystal structure of class 1, which is green and blue here, they align really, really well. So structure-wise, CD1 looks almost exactly like class 1. The only difference is that the binding groove is sort of longer and skinnier. And that's really so that it can fit those long fatty acid tails. And so you should be able to see this here. Yeah, so here is the lipid binding groove. We've got sort of the head group at the top, and then the long fatty acid tails go down deep in here. You can see this here where these tails are going into the back. And you can also see there are usually these little like tracks for different fatty acid tails to bind in CD1. So structure-wise, CD1 basically looks identical to class 1. If we actually look at how CD1 works in terms of where it gets the lipids and which parts of the cell it goes through and things, it does exactly the same thing as class 2. And so CD1 can come into the endocytic or the biosynthetic secretory pathway. It will pick up some random lipids there, and then it will present them. So the process is really the same as the process for class 2. You might notice that on a couple of the slides you've seen CD1 listed as like CD1A, CD1B, things like that. I am not going there at all. The answer is that, in fact, in humans, we have multiple CD1s. Most other animals seem to only have one, including mice. And so we pretty much just know how like the one in mouse works. And we don't totally know like what the differences are between the ones in humans. There's argument. I don't even care that we get into that argument. And the T cells that recognize CD1, or sometimes we say the T cells that are restricted by CD1, are called NK T cells. So they're T cells, but they also have some properties of this other cell type that we'll learn about later called NK cells. They're not NK cells themselves. They just have some similar properties. They are T cells, but they're weirdo T cells. So like they're their own thing called NK T cells. So you can notice this aspect of lipid presentation, and this is going to turn on a different group of T cells, not the CD4s or the CD8s, now a different group called NK T cells. All right, so the other thing that I want to briefly mention to you, because this sort of segues us into Friday, and again, people just find it fun and cool, is about evasion. So MHC class 1 and class 2, but we're really going to talk mostly about class 1 here, is really, really, really important for making a CD8 response to viruses. And that really does a lot to control those viruses. Viruses do not like MHC class 1. MHC class 1 is their big time enemy. As a result, viruses can do a lot to try to avoid MHC class 1. In the paper that we're reading for Friday, we're going to see a situation where we're looking at a virus that has a relatively small genome, and it's going to try to use mutations to avoid MHC class 1. But there are other types of viruses that try to evade MHC class 1 in other ways. Some of them are not little small viruses that just try to mutate. Some of them do other fun things. And all of the other examples I'm going to show you are viruses that are pretty big, and then I'm going to bring this back to make a bigger point. So one virus is Epstein-Barr. This is the causative agent of mono. Epstein-Barr has this protein called EBNA1 that it makes a ton of. And so you could imagine it would be like on lots of MHC turning on lots of T cells. EBNA1 has these long stretches of glycines and alanines, and it can't actually go into the proteasome. It has a sequence that basically makes it unable to be degraded by the proteasome. And so it can't get proteolyzed, and its peptides can never get presented. So basically, you can imagine this protein is hiding from MHC class 1. It's pretty sneaky. The old standbys are these two proteins called US6 and ICP47. They're from either viruses called HCMV or HSV. You don't need to know which viruses or any of that. Just think about the mechanism is going to tell us something important. And these viruses actually make little proteins. And if you look at the structure of these proteins, they actually look like stoppers that function to stop up TAP. And so they plug TAP. So no peptides can go through TAP. You can see one of them plugs it on one side, one of them plugs it on the other side. Either way, the idea is that we completely plug TAP. And these were the first ones that were found. They're pretty cool. Adenovirus, E19, actually takes tapicin, the chaperone we had in class 1, and rips it out of the ER membrane and throws it in the proteasome. So it just steals this thing. It steals the chaperone and stops binding. Murine gamma herpes virus, MK3, takes the whole MHC molecule out of the ER, and it's just like, nope, you're not filling that. You can't it's going to just destroy the whole MHC molecule. There are even other viruses that bind to the MHC when it's on the surface of the cell and pull it off the surface of the cell. And what I want you to see here is that basically all of my 6 steps have some virus that messes with it. There's some virus that tries to mess with MHC class 1 presentation in all of these situations. Some of them, like the EBNA1 situation, makes it so that the protein can't get proteolyzed, but all the rest of this process works. But all of these other ones are pretty dramatic. Like if you block TAP, not only are you going to make it so that your own personal virus proteins can't get on MHC class 1, you're going to make it so self proteins can't get on MHC class 1 either. Similarly, if you pull MHC out of the membrane here or here, you're stopping presentation of self proteins and you're stopping presentation of non-self proteins. And so what I want you to see is that some of these things would completely stop presentation of all self proteins. Your body has a way to actually check are self proteins being presented. This is such a common strategy for so many viruses that you have actually evolved a way to check, is there MHC on the surface of my cell at all? If there's no MHC on the MHC class 1 on the surface of the cell, that means some virus is mucking it up, is trying to hide. And so this presentation of self peptides is happening all the time. And it is actually a key way that the cells are kind of indicating good homeostasis to the immune system to say, no, I'm doing okay. Nothing has messed up my class 1 presentation. And so this self presentation is something that you need to be aware of as happening all the time. And if a cell ever stops presenting on class 1, that's a huge problem. Your cells right now, when you're not sick, should be presenting self peptides on class 1 all the time to show that they are just doing cell processes well. And if they're not, then huge issues come in. And part of this evolution is because of all these viruses that want to muck with this so badly. And we'll see another way that a virus can mess with it when I'm talking about our paper on Friday. So I'll see you guys then.